This tutorial will cover how to prove generated using computational algorithms in the current annotation of the maize genome using the sequence of known genes. For the purposes of this example, we will be using a gene called sugary1, which is involved in the conversion of sugars into starches in maize kernels, and mutations of which are involved in making sweet corn, which obviously keeps the sugars rather than converting them all into starches. To begin with, we need to find a sequence of sugary1. We will simply go to GenBank and search for sugary1. And we're going to use this one, which was actually, as you can see, part of the original cloning of the sugary1 gene. So it's important to note this ID number, so let's save that. And of course we also want the FASTA formatted sequence of this coding region. So with these two pieces of information, we're now ready to go into CoGe. We'll want to start with CoGe Blast. So using CoGe Blast, first we want to specify the organism we're interested in, which is of course maize. And we're interested in the version 2 of the genome, and let's use the working set gene annotations just to be sure that if there's any gene model at all, we'll be able to capture it. Now all we do is enter in the cloned sequence. As you can see, it's already set to BLASTM, so that's fine. Since this is comparing a genome and genes from the same species, we can set the e-value quite high and then just let this run. So as you can see, there are a wide range of hits. Clicking on HSP allows us to see that many of these hits all match to the same gene. So now let's capture this gene name. So let's capture the name of this gene. And now we can move on to the second step, which is of course GEVO. So if we go to GEVO, let's enter the name of that gene we've identified as the potential sugary1 ortholog, or the potential name of sugary1 in maize. And now for the second sequence, we can simply enter the GenBank ID that we saved earlier. And then just click Run Analysis. Alright, as you can see, this is a little confusing in that there are large segments, including exons and introns, that are all grouped together, and that's because by default GEVO uses an algorithm called BLAST-C, which is designed to span large gaps. Since we're actually interested in how well this cDNA matches to only the annotated exons of this potential sugary1 gene, we're going to switch to BLAST-N and rerun the analysis. Now, as you can see, I'm going to hold down Shift and click so you can see all the exons at once. There is a very good correspondence of each part of the cDNA to each individual exon of the annotated gene. So in this case, we can have high confidence that this gene annotated by maizesequence.org is in fact a good representation of the sugary1 gene that was cloned back in 1995. Obviously, you and I have seen this, so we're convinced of it. Let's say you wanted to save this and show it to a friend who was not convinced that the sugary1 gene model in the maize genome was correctly annotated. To do that, you simply take this link and copy it and save that information so that you can send this link to anyone and they'll be able to regenerate the exact same analysis that you have with the same gene, the same sequence from GenBank, and of course the switch to the BLAST-N algorithm.